The cell cycle describes the stages of cell division. It is often depicted as a circle separated into segments, with each segment representing a phase of the cycle. There are 4 phases plus 1 special state for resting cells. The first is gap phase 1, or G1. This phase begins after the completion of cell division. Cells increase in size and begin to synthesize RNA and protein in preparation for DNA replication. This is followed by synthesis phase, or S phase, which is the period during which DNA is replicated. Gap phase 2, or G2, begins when DNA synthesis is complete. During this phase, cells continue to grow until they have doubled in size and have enough cellular material for 2 cells. They then transition into mitosis, or M phase, during which time the cell divides, creating 2 daughter cells. The diagram sometimes has an additional phase, resting phase, or G0. A cell enters G0 by failing to pass a checkpoint in G1 that determines if a set of conditions needed to proceed with the cell cycle are met. Cells enter resting phase for a number of reasons, and transition into this phase can either be reversible or irreversible. Cells in G0 may function normally, but are not actively synthesizing the materials needed to proceed through the cell cycle. There are also checkpoints at different places along the cycle where the cellular machinery makes sure that certain conditions have been met before proceeding to subsequent phases of the cycle. By showing growth during the cell cycle as a chart, we can see where different types of growth occurs. RNA and protein concentrations increase throughout G1, S, and G2 phases. As a result, the amount of RNA and protein doubles from the beginning of G1 to the end of G2. These amounts then drop back down as the cell divides during M phase, with mitosis and cytokinesis occurring. Contrast this with DNA, which also doubles over the cycle, but in the case of DNA, all the synthesis occurs during the S phase. In a normally dividing cell, the cycle begins at G1 right after it is formed by the division of its parent cell. As the cell enters G1, the nucleus contains a single copy of its genome. The chromosomes are uncoiled to facilitate DNA, RNA, and protein synthesis. The cell increases in volume as RNA, protein, and organelles are synthesized. And during G1, the proteins and enzymes required for DNA synthesis are made in anticipation of entering S phase. The primary checkpoint for the whole cycle occurs towards the end of G1. Active growth during G1 signals that conditions are good for DNA replication. Initiation of DNA replication marks the transition to S phase. Once the cell transitions from G1 to S, it is committed to cell division. In S phase, all other cellular functions continue. RNA concentrations, protein concentrations, and cell volumes increase as the amount of DNA doubles. Transition into G2 occurs at the completion of DNA synthesis. In G2, the cell finishes doubling in size and synthesizes enough organelles and other cellular material to support 2 cells. Cellular components begin to move to the proper side of the cell in anticipation of cell division. This includes the centrioles, which will pull sister chromosomes to opposite sides of the cell during mitosis. The transition from G2 to M occurs as DNA recondenses to form the characteristic X-shaped chromosomes. As the cell proceeds with mitosis, spindle fibers form between the centrioles and each chromosome. The nuclear membrane breaks down, and the chromosomes line up in the middle of the cell along the mitotic plate. The cell begins to elongate, and sister chromatids are pulled to opposite ends of the cell. At the end of mitosis, nuclear membranes form around each set of chromatids, and cytokinesis occurs, resulting in 2 cells, each with half the cellular material of the parent cell. Each cell then begins the cycle again, starting with gap phase 1.